Science Uncut, presented by the Volkswagen Foundation.
I'd like to thank the organizers for giving us the opportunity to discuss some of our personal views as junior scientists on this topic. We have collaborated on this a little bit and exchanged views because we need to kind of add something new and we don't want to be too repetitive. I only finished my postdoc just about 3 years ago, and have only now, or 3 years ago, started my group as an independent group leader back in the UK. I thought, first of all, I should just give a little bit of context as to the types of experiments and work that we do to give you an idea of where I'm coming from. So my lab is interested in the fundamental replication cycle of influenza viruses, and in particular how one virulence factor of influenza virus manipulates the host cell environment. And we do gain-of-function experiments to try and address this. So this NS1 virulence factor is a very potent antagonist of human immunity. During infection, it manipulates many host cell processes in order for the virus to replicate efficiently. And most human-adapted virus NS1 proteins are very efficient at this process. However, there are a number of exceptions that we've started to learn about and characterize from a fundamental biology perspective in the last few years. So the original H5N1 influenza virus that started to emerge into humans in 1997, the NS1 protein of this virus lacked the ability to efficiently antagonize human innate immunity. And this virus, or other H5N1 viruses that subsequently came back into humans in 2004, naturally acquired this potential, and this made these viruses more virulent. My lab, in the past, when I was working with Adolfo Garcia Sastre, and now, characterizes NS1 proteins from novel zoonotic sources when these viruses enter humans. So when the 2009 pandemic began, the swine flu pandemic, and this virus emerged from a pig population, it was an unexpected in vitro finding that this NS1 protein lacked the ability to really efficiently antagonize host innate immunity. And we could characterize this in vitro, but then take it forward into a gain-of-function experiment to test the hypothesis of what would happen if this virus acquired this potential. And we could actually show that acquisition of this property in the circulating pandemic virus would not increase virulence of this virus. So we could define the amino acid motifs that were responsible for this function. And this could tell us that if this property was acquired in this emerging virus or in the virus that was circulating, this would not necessarily increase or impact the virus disease in humans. However, this completely contrasts with a different virus background. So in H7N9, another virus that has spilled over from an avian source, this avian NS1 lacks this particular property to efficiently antagonize host innate immunity. Again, we could predict this because of the basic research that we'd done previously, and we could identify a motif that may be responsible for this. And again, using, in this case, quite heavily risk-mitigated strategies where we did not use the external H7 or N9 subtypes on the virus, but we were only interested in the internal genes of this H7N9 virus, we could show that if this virus naturally acquired this human adaptive ability, it may increase the virulence of this particular virus. So these kinds of studies have implications for our understanding of what virulence motifs we might be worried about in natural acquisition of these viruses. But also it underscores that whatever impact we see in one strain does not always transfer to the other strain, and different strains have different consequences. And actually these consequences are unexpected. We did not expect that this acquisition or gain of function in the 2009 pandemic would potentially attenuate this virus. So that is just the context of what my lab works on. I now want to just give just two simple slides on some of the thoughts that I've had or that we've had during this process or during this conference that we've attended. And firstly, I just want to make the point or to clarify in everybody's minds, which I'm sure that everyone understands this point, but in some of the discussions I feel that this has been lost, that gain-of-function research actually encompasses a wide range of experimental setups and is not limited to studies on increasing virulence or transmission. There's a lot more to gain-of-function than just these two aspects. In fact, most virology experiments to some extent will include a gain-of-function aspect that we may not be concerned about. So the earliest flu experiments were probably passaging the newly isolated virus in mouse brains to try and get a mouse model for this virus. Our earliest mouse-adapted viruses are, in a sense, a gain-of-function type of experiment. When we adapt a virus that we want to generate as a vaccine to a cell culture or an egg culture system, we are giving it a new property. That is what gain-of-function is. And when we can engineer influenza viruses to incorporate new items into them. In many instances, we put in reporter genes, such as a green protein, so that the virus will fluoresce as the virus replicates. This is a gain-of-function type experiment that you would consider not to be threatening, but is incredibly useful for us to understand the cell biology of these viruses, because we can rapidly monitor how particular viruses are replicating and then identify host factors that are regulating these processes. So I just wanted to bring the point that in this discussion, some of the boundaries between the words Dirk and gain of function appear to me to have been slightly muddied. And I would like to suggest that we should be really very specific about what we are worried about or what types of experiments we might be worried about, and not term everything gain of function and not seek to throw out all gain of function experiments. And I know that not everyone thinks like that. In fact, everyone agrees that all gain of function is very different, but just to keep that clarified. And then I suppose this is my final brief slide, and we've come up with this together, Silke and I. What are the potential impacts of some of this debate on junior scientists? I'm sure I don't speak for all junior scientists at all, but the uncertainty that's been caused by this debate and some of the pauses that have been either self-put in or are now being mandated by governments are going to have impact on people with careers that are at their very early stage and are actually under really tight pressures because they are early on. So new PIs such as myself have very short-term grants. We need to prove ourselves very early on in order to confirm tenure. And pauses that will delay our work or stop our work are not compatible with grant deadlines and reporting deadlines. Whether this is an issue with the debate or whether this is something that needs to be brought up with funding bodies is another question. And of course, students and postdocs who are on very short-term contracts and need to publish their work and are told to stop their work immediately or move on to another project, it's very difficult for them to complete their theses or to write up a coherent body of work. And this is an issue and will be an issue for people working in this field. There has also been quite some debate about restricting many of these types of gain of function or certain experiments or Dirk experiments to particular facilities or well-known and well-characterized laboratory heads. And we feel that this may impact the independence of new investigators. Silke and I have been trained extremely well at Mount Sinai School of Medicine in working in these containment labs. At Glasgow and also in Zurich, we have excellent facilities for containing particular pathogens, but are we going to be negatively impacted as junior PIs and have to be under the auspices of another larger PI? Then, of course, this debate, will it impact or harm the attractiveness of the field in general to incoming people? Or even people who are already here who think that this is going to be quite a tough sell to try and get our science across and try to justify ourselves a lot more. And this leads on to just a very, very final point that I feel has come up in the last couple of days, just some kind of uneasiness that I feel that this debate went, at least yesterday or maybe in the past, that a lot of the work that our colleagues are doing, our senior colleagues, and perhaps our science, is being linked now to aspects that are contravening biological weapons conventions or a part of weapons of mass destruction. And actually, this linkage, although in law may be a way that there is a basis for linking it in law, is actually, I'm sure, incredibly demoralizing for people and for scientists to think that our research, where we are trying to help and trying to understand the mechanism of how these viruses are working and how they're causing disease, to be linked with these kinds of things is certainly going to be quite demoralizing. So that's all the opinion that I wanted to put forward, which of course is just personal opinion, and if anything else, just to open up the discussion and to start to ask some questions. I'll now move over to Silke.
I would also like to thank the organizers for the invitation to this meeting and the chance to present my perspective. So I will also give you a very brief summary of the type of work that I'm doing in my group in Zurich. So we are interested in really the cell biology of influenza viruses. So what we do is we try to understand how the virus and host interact. It's a very small virus that relies on the host cell for its entire replication cycle. And we try to shed some light on this interplay. And we focus on virus entry into a host cell. And in this area, we try to define the cellular proteins that the virus relies on, that it uses. And we try to understand the mechanism behind this interaction. And the long-term or overall goal of these things is to maybe use some of this information to develop new antivirals. If we have a host factor that is absolutely essential for the virus and we can drug target it, we might have a chance to develop new antivirals. A second part of my research also looks at the other side. So these would be host factors that can actually block or restrict the virus. And we feel that from studying these kinds of things, we can also learn weaknesses, weak spots of the virus, and maybe eventually develop strategies to block the virus. So this is where I'm coming from. And my experiments involve influenza viruses, wild-type and genetically modified ones at biosafety level 2 and 3. My experiments currently do not contain any of the DIRC experiments. So I do not have a direct vested benefit from these experiments, yet I feel quite strongly that these experiments are extremely important and they are an important tool to answer a very important questions. And I would like to make 3 brief points. So first, I would like to take you briefly through the process we're going through, or I have been going through before actually doing a project. And so we first need to decide whether the experiments we're thinking about are scientifically justified. What question do we want to answer, and are these the right experiments? And we then do a risk assessment, and we think about risk mitigation strategies. What is the appropriate containment? So we already do all these things. And we then have a quite rigorous process of getting these things approved. So we discuss it with colleagues, and then I, for example, need to discuss it and propose it to my institutional biosafety committee and get the approval. And then I need to go to the national level and get these things approved. And then once that has been done, the funding body will again review these processes and see if that makes sense before I can do anything. And even if I get approval, this is not where the work ends. I then have to train and select the personnel and make sure that these people are trained on a regular basis. So I need to take responsibility. And so this is to say there is already a lot of thought and a lot of work going into these aspects of biosafety. Well, obviously, I don't want to say everything is perfect, there's no problem. Because one area where I really see a need for improvement is the need to really communicate our decision process. I think this is something where we're not as good as we should be. Why are we doing these experiments? Why are we doing the risk mitigation strategies or don't? And why do we choose the containment level? What are our thought processes and how do we come to our decisions? So that, I think, is a very important point where we as the scientists need to get better. And the second brief point I would like to make is regarding this second point, the third point, the risk mitigation strategies. So it has been suggested a number of times that we could answer this question of what makes a virus transmissible with other approaches. We could use seasonal strains, we could use loss of function rather than gain of function. And I have to say I have some trouble following these arguments because this is not a new question. The question has been in the field for quite a few years, and a lot of very smart people have put a lot of time and effort into thinking about how to answer this key question. And a lot of experiments have been done using alternative strategies. But what I feel is a consensus in the field is, we have learned things from these experiments, but is not enough to really understand and answer this key question. I obviously cannot speak for the whole field, but in my opinion and my experience from discussion with colleagues, we do agree that these are the experiments to answer that question. And I think knowing this is a key question and knowing how we could get answers and then not to do it, that does pose risks as well. Not just doing it, also not doing it does pose a risk. And I think this risk should also be included in the discussion. And then my last point is really very brief because we spent a lot of time discussing that yesterday. So you heard that from the German Ethics Council there has been this recommendation and this statement. And those were the 5 major recommendations. I only want to briefly mention the one with the DIRC committee. So they suggest to establish this DIRC committee, and they want the experiments to be approved or reviewed by this committee. And then you also heard that the Society for Virology of the German-speaking countries has also released a statement, and they agree with large parts of it and say, yes, there are very important points in there that we should follow. However, they feel very strongly, and this is again something that is my personal opinion and that has come across from lots of discussions with colleagues in Germany, there needs to be a practical solution. Having another committee that is really tough, and I'm not sure that's a realistic solution. So if we could incorporate this in an existing committee, like the ZKBS, which currently does analyze the biosafety issues, I think it would make a lot of sense to combine these things and really aim to find a realistic solution. So those were the brief points, and just remember, it's my personal opinion, and I just wanted to give you my perspective on this. So, thank you.
The floor is now open for comment or question from the audience, and well, I will begin. So I would like to begin actually by asking Ben and Silke, you heard what David also said about the responsibilities of scientists. Did you feel in your preparation and training that you had adequate engagement, opportunity to think through also these levels of social responsibility in addition to the techniques and skills that are necessary and obviously take up all of the effort to become prepared to be practicing scientists?
I don't think so. I have had very good training regarding biosafety and these kinds of things, and it has become clear that I would have to take responsibility for these things, but how to communicate it, that is quite new. So currently, there are not that many influenza labs in Switzerland, so if the newspapers have a question, they end up calling me. And in the beginning, that was quite a strange position. I did not expect it, and I wasn't aware that this is part of my job, so I'm kind of learning as I go along. But yeah, I have been trained. Yeah.
Ben, do you want to add a word?
Yeah, I mean, I think I would agree with that. That as a junior scientist working in labs, you think of the benefits of what you're doing and the health implications of the questions that you're trying to answer. And then it is only I mean, we didn't do at the time any dual-use research during those times, but you know, in later times now, thinking beyond that and the other side of the coin is something that we now consider, and it's been a steep learning curve, and it is something that can be improved, and something that already I know Silke is starting in Zurich with her students is getting them involved in the process, and in Switzerland you already have this Dirk meeting, right, for your students. So it is something that is starting already, and we are taking seriously.
Great, thank you.